So hello everyone, and thank you for joining us for our May GLASE webinar. My name is Haley Hawley. I'm the Extension Specialist with GLASE, or the Greenhouse Lighting and Systems Engineering Consortium. And before we start our webinar today, as usual, I would like to thank our current GLASE industry and CEA members for their support. It's because of them that we can promote these activities and offer them to a wide range of audience, such as yourself. So for our presentation today, remember that there is a Q&A box at the bottom of your Zoom screen, and you can use that to ask questions. We will save some time at the end of this presentation so that we can answer questions from the audience. So be sure to put your questions in there as they come up. And today we will be hearing from Dr. Neil Mattson from Cornell University and Dr. Ricardo Hernandez from North Carolina State University about the response of cannabinoid hemp to light quantity and quality. Dr. Mattson is a professor and greenhouse extension specialist in the School of Integrated Plant Sciences at Cornell University. He's the director of Cornell's Controlled Environment Agriculture program, and his research is focused on optimizing crop performance and resource use efficiency of vegetables, small fruits, and herbs. He also has research interests in strategies to reduce energy use and CEA lighting through LED adoption and intelligent light controls, sustainable fertilization practices, and development of new CEA crops. Dr. Hernandez is a faculty member in the Department of Horticultural Sciences and Controlled Environment Horticulture at North Carolina State University. He's also the director of the NCSU CE Coalition, which aims to develop controlled environment agriculture as an economically and environmentally sustainable option for agricultural practices by performing evidence-based transformative research. Dr. Hernandez is also a co-founder of a vertical farm startup and a startup focusing on developing CEA technologies for the propagation of plants indoors. So with that, I'm going to turn things over to Dr. Mattson and Dr. Hernandez.
Thank you very much, Haley. I'm gonna go first here, and let me share my screen with you. And if you can, please confirm that you're seeing the correct slides here. Haley, can you see the correct slides?
I'm not seeing a shared screen right now.
Yep, give me 1 second. Let me try that again. How about now?
Looks good.
Thank you very much. Well, thank you, everybody. Thank you for your time. I know you are all under a busy schedule, and this presentation is going to be led by me and Dr. Mattson. And because we're working collaborative in this project, Cornell University and NC State University. I'd like to first thank our research sponsors that have made this research possible on the NC State side of things. We have the Hemp Research Consortium, also the Foundation for Food and Agricultural Research, Current Lighting Systems, and Hawthorne Horticultural Solutions. And also I'd like to give full credit to the lead scientist on the research that I'm going to present. Is my PhD student, now PhD candidate, Cristian Collado, and he deserves all the credit for the information, but I get to talk about it. So thank you, Cristian. So let's start with basic measurements. And probably most of you, if you have followed GLASE presentations, you know the importance of light. So this is just going to be a very brief reminder. And one unit that I want to talk about is your daily light integral, which is the cumulative amount of photons between 400 and now 750 nanometers received per an area for the entire day. The area is typically the meter. And this measurement is so important because the amount of light received per area is directly correlated to the potential yield of the plant. And that DLI changes depending on where you're located. And here you have two types of measurements. You have the outside DLI and the calculated inside daily light integral inside of a greenhouse. And this is just around 30% reduction from the outside based on some numbers. But this is what you can expect inside the greenhouse. And that depends on the season, right? So this is the annual average. But depending where you're located and depending on the season and the location, that can change. This is now January, a very dark year, right? So light inside the greenhouse is around 7 to 11 moles. And in areas with a lot of light here in Arizona, it's around 20 moles. You can be in areas where you have lots of light that you need to actually do deploy green shade cloths to reduce the amount of light. And this is the example of those daily light integrals in June, in the middle of the summer, right? So, and typically it's important to know your daily light integral because that can be your potential for yield. Here you have, again, the average inside light integral in the graph over here, depending on your location, right? So you have Arkansas all the way to Tucson, Arizona, which has a lot of light. And typically it's recommended to have between 30 and 35 moles per meter square per day average for tomato and cucumber optimization. Typically for young plants and vegetables and leafy greens, it's typically around 13 to 17 moles per meter square per day. In Southern Arizona, you know, the average inside the greenhouse is around 20 moles per meter square per day. And, you know, and I picked Arizona here, Southern Arizona, because it's one of the places with the most light in the US, right? And in the Netherlands, for example, they may have up to 2 to 4 moles per meter square per day average in the winter inside the greenhouse or less, right? So knowing these measurements can help you understand your potential yield. If I draw a line in here, you can see here that inside the greenhouse, where there is only a short season here between June and July, we have enough to maximize tomato growth here. However, during this same season, it is necessary to deploy a shade cloth to reduce the overall radiation. And it's not because you have too much light for photosynthesis, but instead it's because there is too much energy, too much heat that is getting the crop temperature above optimal. So we're always battling to get more light. So therefore, knowing your daily light integral is very, very key. And if you know your daily light integral of every day, a better measurement that will predict growth is actually your cumulative light, right? Which is the DLI of today, plus the DLI of tomorrow, plus the DLI of every day until the end of your season. And that's so important because you can actually calculate how efficient is your specific crop to light, right? So you need your cumulative yield per area, and growers are typically very good at knowing how many kilograms of tomato, how many heads of lettuce, how many pounds or kilograms of cannabis flowers they can get per meter square area. This is a unit that growers are very familiar with. Another unit that growers should be familiar with is the cumulative light per area, right? Moles of light per meter square per day of every day. So if you have those two numbers, you can plot the numbers in a graph like this. And all of these, I recommend you this publication by one of our colleagues that actually walks you through some of these exercises, you know, and I extracted this data from this publication, Dr. Kubota from Ohio State University. But this is actual data from a tomato greenhouse. Here you have the cumulative yield in kilograms per meter square, and here you have your cumulative amount of light for the entire season. And if you draw a line in there, you can see that is linearly correlated the amount of light to the yield. And if you ask for an equation for that line, basically here, you can get 0.013 kilograms of tomato per every meter square area for every 1 mole of light, right? So just remember that number. This is kilograms, but if we change that to grams, it will be 14 grams of tomatoes for every mole of light, right? So you can do that for many crops. And I'm going to leave this just as a reference. I don't have to go through these numbers for the entire table, but you can see how different crops and their efficiency is different based on light. You can see tomato, this is the 14 grams that I showed you just now, grams per mole. Cherry tomato is a little less efficient, but also you get more wholesale price per kilogram. Lettuce is just less efficient than your tomato on the vine, but you get a lot more per kilogram of the lettuce. And strawberry, strawberry is a little lazy when you're talking about light. You can compare these 2.1 grams of strawberries per mole of light versus 14 grams of strawberry per mole of light you're doing TOV. But you also get a little more money for strawberries. So if you compare all of that, you can get how much money you get for every mole of light invested for every meter square. I'm going to put a pause on this table because we'll come back to it after the next few slides. So now let's jump, now that we discussed about lighting, let's jump about supplemental lighting for cannabis growth in the greenhouse. So cannabis has different production stages. You have your nursery, and here you get cuttings, and these cuttings need to get rooted, and those roots and those rooted plugs now become a new plant. And then you have the vegetative stage, and that ranges on time. And this specific experiment was 3 weeks and 1 day. And we kept those on the log photo period to prevent flowering. And then you have your flowering induction, which also depends on your crop. And the specific example was 7 weeks and 1 day. Typically, I hear growers keep them under short photo period for flowering for up to 8 weeks. All right, so the objective of this was to study the impact of this supplemental lighting in both the vegetative and the flowering stage of cannabis. Here you can see the different light treatments, and basically the green portion is when it's in vegetative under long photoperiod. The blue portion is under flowering under short photoperiod. So we have the top line here is the line supplemental light intensity with LED lights, which is in addition to the sun of the supplemental light intensity of 150 all the way to 700. But we also wanted to know what would be the plant response if we had different intensities while the plants were in vegetative versus flowering. So we actually kept the treatments of vegetative light intensities different, while all of them, so it was 3 weeks, are under 150 and then move for 8 weeks to 700. We've seen some growers that actually keep lower light intensity at the vegetative stage because they make the assumption that the plant is younger, so it may need less light, right? So we wanted to test that hypothesis. Can you start it on the lower light and then move it on the high light and see the impact? So here you have a greenhouse with the flowers here, harvest day, and there's different supplemental lighting treatments. And here you have a greenhouse with the vegetative stage of the plant, shorter, more compact. We collected many things, but what I want to focus on is on the vegetative output and also on the flowering output. So here in this spot, you have the main stem in orange, and then you have the blue lines, which are called primary branches, right? Those are branches coming out of the main stem. And then these green lines are called secondary branches, which are branches that come out of the primary branch. And so we quantify all the yield. This is important because if you run in a nursery, every of these single secondary branches will become a new cutting that will then become a new rooted cutting that you can sell into the marketplace for the vegetative part. So what we found is a direct correlation between cumulative light and branches per meter square. So the higher the light intensity, supplemental light intensity, you have here 700 micromoles. If you give, in this specific experiment, the average DLI was 52 moles per meter square per day. Remember, I said to maximize tomato, I suggested 35 based on literature. Well, here we're giving out to up to 52 because it was the 700 plus the sun. So the sun provided around 17% of the light, and around 83% of the light came from the supplemental LED. And you can see those partitioning depending on the supplemental light intensity. For example, if you gave 150 micromoles, around 50% came from the LED, and around 50% came from the sun. But it didn't matter. It didn't matter the percentage that came from the sun or the LED. The only thing it mattered in terms of branches per meter square is that more light, more branches, right? So we focus on this number here. We can get around half a cutting, half a branch for every mole of light. And we'll come back to that now. And the main effect was in the secondary branches. You can see the primary branches, we have very little effect, right? So these blue ones is more a developmental response and not really a biomass response. So most of the plants were the same height and have around the same number of primary branches, but depending on how much light, you were able to get a lot more of these green lines, which are the secondary branches. You can see here an example where the plants are all the same height independently of the light intensity, because that was mainly driven by the internal length between the primary branch. And since I mentioned that primary branches were around the same, they all have the same height. But the higher the light intensity, you had a lot more foliage, and then therefore a lot more secondary branch. So how about the actual flowering stage? We also measure lots of things, but we're going to talk about the yield and some cannabinoid and terpene composition. Right, so we go back to those same experiments, right? But now, while in the vegetative, we only have the 150, 300, 500, and 700 intensities. Now we have all the static intensities and also the dynamic intensities as results. So here is the result. So this is flower dry mass, which is grams of dry mass with 10% moisture, right? So that's typically what you find on cannabis flower biomass, 10% moisture per meter square, grams of biomass per meter square. And this is the total amount of light for the total 72 days. The black dot is the constant light where it was, these treatments up here, always at the same light intensity the whole time. And the gray dot is the cumulative light on the treatments that actually were changed like this, right? This 700 is the same. That's why only you see one dot on it. And here you see the percentages, right? So now we added the plant that went from 150 to 700. It had 26% sun and 74% LED, which is almost the same that the plant was at 500 the whole time, which was 22% sun and 78% LED. And these 2 points right here are these 2 points right. So basically, what I'm trying to tell you with this line is that it didn't matter where, at what time you gave the light to the plant, either vegetative or flowering. The only thing that matters is the cumulative amount of light given in the entire growing cycle. In other words, if you give less light during the vegetative, then that will be penalized in your flower yield. All right, so the plant was very, very efficient at actually getting photons of light. These are the plants at the end of the experiment. You see the scale is around 51 centimeters. They look small here, but those are 4-gallon pots, so they're quite big. These are the ones that actually move from lower to higher, and these are the ones that stay on the constant light intensity. Right? So what happened with the percent CBD and the percent cannabinoids and the different light intensities? Well, we found that if you grow plants under very high light, or if you grow plants under very low light, the percent CBD and cannabinoids did not change. The percent per dry mass base. This means if you take one leaf and you take dry mass of that leaf and you have the same amount of leaf and you check the percent CBD and the same amount of leaf, and depending where the leaf is coming from, that percentage is the same. So there was no penalty by increasing the light intensity on the percent CBD, but also there was no benefit in terms of percentages of increasing, right? So you can feel safe that the quality and this percent will be most based on the genetics and not on the light strategy. We also plotted the total CBD to the total THC. And whatever the main thing this is saying is that if you're able to increase your CBD percent based on light, you had a cultivar that was not CBD based, but it was different THC based, there will be a similar response because they're highly correlated. So summary points, we have 1 mole of light can produce 0.21 grams of dry flower per meter square per growing area, right? 1 mole of light, 0.21 grams. We did some economical analysis that I didn't include here because of the time, but basically for CBD dry flower, you can make around $22 to $107 per meter square per growing area, depending on light intensity. All right, so very big range. You add 150 micromoles, or if you add, 700 micromoles. THC dry flower is a lot higher per meter square, depending on the area. These are the assumptions for these comparisons. We assume only half of the flowers were markable viable. We assume that all light is sole source, so actually the cost of lighting that greenhouse was higher than the supplemental lighting. We assume this CBD flower market is a little different now, and we assume this THC flower market cost, and we assume a fixture efficiency of 3.1 micromoles per joule and a 0.09 kilowatt hour electricity rate. So remember that table that I was talking about earlier in the presentation? Here you have your tomato, 14 grams per mole of light. Now, if you go back to one of those equations for the cannabis flower at 10% moisture, I just told you it was 0.21 grams per mole of light. All right, now this is dry grams. This is fresh. It's actually a fresh tomato. It's a dry flower. If you go back to the cutting, which is the secondary branching, we get around 0.4 cuttings for every mole of light. Now, we compare that economically speaking, those cuttings are right now, they're typically sold around $2 per cutting. In Colorado, you know, we pick the best case for flower here is around $1,563 per kilogram of flower, right? And if you do the math here, it means the profit per mole of light, 72 cents per mole of light in a cutting, or 32 cents per mole of light on a dry flower of cannabis. If you compare that to perhaps lettuce, which has the most response to light, you lettuce, very profitable crop to put supplemental lighting on, you get $0.041 per every mole of light per every meter square. You see that difference is pretty crazy, and this is mainly driven by the market prices. So you can change this number depending on the market price, and you get better understanding of the market. So we talked about the actual plant that it will be a mother plant to produce cuttings or a plant that will produce actually flower. I want to teach a little bit about into actually the clone, right? Which is the cutting. And in order to get the cutting, you have the plant, the mother plant, you get the cutting. These are the secondary branches. And then you root those cuttings to produce these plugs. Right? So we wanted to see if we could manipulate the light to actually produce those plugs. So we actually rooted these plugs and a different light intensity treatment in a climate controlled chamber, you know, being very careful on measuring that photon flux, right? And we measured the rooting after 14 days. Right? So what we found here is that this is over here, the light intensity provided during rooting, and this is the root dry mass in grams, right? So we found an increase in root dry mass, you know, in kind of plateau around 400 micromoles per mole. And 400 micromoles per meter square per second light intensity. I was quite surprised to see this response because typically we use a lot lower light to root ornamental plants. We also use a lot lower light, even lower than 100, to heal grafted plants and to root or rooting cuttings. But here you can see with a good control of the humidity, even very high light of 300 when the plant had no roots, it still increased the root development and growth of the plant. And also includes the shoot fresh mass linearly. And the shoot fresh mass, shoot fresh mass and shoot dry mass as well, so growth of the plant linearly. While the plants were there for only 14 days in this system, right? So what happened then? So after the 14 days, Christian took these plants and pot them and put them in the greenhouse for another 14 days to see if there was any carryover effects from the actual light intensity during the rooting part of the cycle. And then we found that, based on the hypothesis that if you started with a bigger plug, and this plug have more biomass to begin with, both in root and shoot, then you end up with a bigger plant at the end of those 14 days. So this is supplemental lighting during the rooting stage also, and then they will move into the greenhouse under the same lighting conditions. And you can see after 14 days being under the same lighting conditions, the plants that were rooted under higher light had higher biomass at the end of those 14 days, 53% greater. Right, so in summary, during rooting, you can give cannabis plants a lot more light if you're able to maintain a good humidity and a good temperature. And the bigger plant that comes out of the rooting environment will have an impact after the rooting environment. So these are the partners of the Controlled Environment Coalition at NC State. And this is the end of my presentation. And this is a QR code so you can access our webpage. And here you have the picture of the lab members. Thank you very much, Neil.
Okay, good afternoon, everyone. Thank you, Ricardo. And a reminder that everyone can use the Q&A to ask questions. Oh, Ricardo, yep, then give me the screen. Very good. So use the Q&A to ask questions, and then we'll take joint Q&A at the end. So we're going to shift gears, and I'm going to talk about one specific, or the effects of light spectrum, and specifically far red radiation. So what is far red? And first I want to thank my master's student, Brett Timmons. He is finishing up his thesis working on the effects of cannabinoid hemp to far red and daily light integral. So far red is wavelengths of light between 700 and 800 nanometers is how we define it broadly. And this is the spectrum of light we get from the sun, with UV light is less than 400, and then blue light is 400 to 500, green light is 500 to 600, and red is 600 to 700. So traditionally we refer to all the light between 400 and 700 nanometers as being photosynthetically active. However, there's a growing body of evidence that that far red light is useful for plants. And so the broader classification of far red is 700 to 800 nanometers. And in terms of plant responses, what we're really interested in is 700 to 750 nanometer. There has been reports going back to the plant physiology literature from even the 1950s that far red light was effective for photosynthesis, so long as it was combined with shorter wavelength light. And there was a great body of work done by Shu-Yang Zhen, who's at Texas A&M now. She was a postdoc at Utah State, and she did her graduate work with Mark Van Eersel at the University of Georgia. And they, through comprehensive experiments, they basically concluded that within bounds, far red light, or 700 to 750 nanometer, is as effective as shorter wavelength light, as long as the shorter wavelengths are also present for photosynthesis. So there's the suggestion that instead of talking about photosynthetically active radiation as being 400 to 700, we should talk about extended PAR, which would be 400 to 750 nanometer. Besides the impact on photosynthesis, we also know that far red light controls the shape of the plant, or the morphology of the plant, and plants see far red light in nature. So typically we can see far red responses which are promoting stem elongation, promoting increased leaf surface area, but maybe thinner leaves, so that the leaves can try to capture more photons that do fall down in shade in nature. And then for some primarily long day plants, far red can also promote flowering. There's a lot of anecdotal evidence of the effects of far red on cannabis, but there are no scientific literature up to this point directly looking at the effects of far red. So we set out to explore this in more detail. And a little bit about far red in nature. So in nature, the sun gives us broad spectrum light, as we talked about. The leaves of the plant mainly absorb or preferentially absorb red and blue, and far red is more likely to transmit through a leaf. So we get more far red and green transmitting through leaves. And then plants that are growing in the shade typically can give us two types of responses, either shade tolerant plants, which grow wider leaf surface area to try to capture the photons of light that do get through, or the shade avoidant plants, which grow taller. And in nature, they're trying to grow taller than their neighbors so that they can reach direct sunlight. So there was not previous work on terms of whether cannabis is shade avoiding or shade tolerant. And so we set about to look at far red at the vegetative and the flowering stage. So typically we have a cycle, and Ricardo had something very similar, where we propagate our cuttings for 3 weeks. Then we grow them under a long day photo period to keep them vegetative for 2 to 3 weeks. And then we grow them under short day photo period for about 8 weeks until they're flowering. And so I'll present results from 3 experiments. The first experiment, we applied far red during the flowering stage. The second experiment, we provided far red during the vegetative stage. And then the last experiment, we provided far red during the vegetative stage. And then we looked to see if there were carryover effects on the flowering stage on flower harvest and cannabinoid concentration. So the first experiment was a pretty straightforward one that was done during the flowering stage. We wanted to determine the effects from 10% far red out of the total photons of light that we delivered on a couple different low THC but high CBD or CBG hemp. And in this experiment, we had 2 different growth chambers. We grew the plants in a common environment for their vegetative stage, and then we applied these far red treatments during the 8-week flowering period. So we had a 12-day photo period. We were a bit constrained with the total light intensity that we could give our plant in these chambers. So we were limited to 17 moles of light, which as Ricardo would say is not sufficient for optimum yield. And the temperature was about 23 degrees Celsius. And then the lighting treatments where we had a control growth chamber that had 400 micromoles of light from white LEDs that were in the chamber. About 2% of that light, or 2% of the photons, were far red. And then in the treatment chambers, we installed far red bars, and then we dialed down the total amount of light that we got from the white LEDs. So we provided 40 micromoles of light from the far red bars, 360 micromoles from the white LEDs. So that total, we had 400 micromoles of EPAR, or broad spectrum light. And you can see the far red peak in that treatment. We repeated this. We did 2 repeated crop cycles where we switched the growth chambers to account for any variability in the growing environment. And then we had plants that were positioned. So we had each row of plants was a block. And then we had 2 plants of Janet's G and 2 plants of TJs, which were the 2 cannabinoid cultivars that we used. Janet's G is a cultivar that is high in CBG, which is its main cannabinoid, and TJ's is high in CBD. So we grew them under these 2 treatments for 8 weeks, and then repeated, and we saw a few trends. So the biggest trend was there was a statistically significant increase in height. So we did see that similar to other plants, we see taller cannabis plants with far red light. For Janet's G, there were some negative effects in that there was a reduction in fresh weight that was significant by about 10%, a reduction in flower dry weight of about 9%, and the stems were thinner as well. So the plant was not quite as sturdy. It was not significant, but for both cultivars, their primary cannabinoid was decreased by about 10% with that higher far red light treatment. So just to put a little bit of a finer point on it, so we have 2 graphs here, the average height at harvest and then the stem diameter. Cultivar Janet's G is on the left, cultivar TJ is on the right. And we did see a significant increase in height for both cultivars with increasing far red. So that increased by 25% for Janet's G, 11% for TJ's, and a reduction in stem diameter for Janet's G. And then flower dry weight, we did see that reduction for Janet's G, but not for TJ's. And for cannabinoid content, there was numerical reduction in CBG for Janet's G and for CBD for TJ's, but those were not statistically significant. So based on that first experiment that we did, far red, that was 10% of the total photons during flowering, led to both cultivars had taller plants with increasing far red light. Janet's G had reduced stem thickness, reduced flower dry weight, and reduced plant fresh weight. So then our next experiment was to look at far red applied just during the vegetative stage. And we changed the experimental design somewhat to do this this threshold experiment. And what we did in this experiment is we went to the growth chamber and we added far red bars that were kind of positioned along the canopy, and we carefully measured the amount of light, both total light and far red light, at each plant point. And so we had higher percentage of far red for the plants that were closer to the bars, and a lower amount of far red for the plants that were farther away. And then the plants that were behind the far red bars were the control plants. So they had about 2 to 3% of far red based on what was in the white LEDs. And then so we had 21% was our highest treatment, down to 4%, and then down to 3%. And then you can see the plants growing here. So we had one chamber with TJs, one with Janet's G, and then we repeated the experiment. So there were some significant findings here, and in this experiment too, we did not grow the plants onto flowering. So we saw a height increase of 25 to 31% for Janet's G and TJ CBD. We saw a dry weight increase of 40% for TJs. It wasn't significant for Janet's G, some indication of an increase in leaf weight, a definite increase in stem weight. So we're seeing more biomass enter the stem, and a slight increase in branching for Janet's G. And so we did these linear regressions where we have far red increasing from 3% all the way up to 22%. And we saw that increase in height at the end of the vegetative stage, which would probably be unwanted by growers. And then that increase in dry stem weight. So for both cultivars, there was this increase in plant height, and then increase in plant dry weight and stem dry weight. For TJs, there was an increase in the plant fresh weight and leaf dry weight. And for Janet's G, we saw increased branching. And so lastly, then we wanted to apply far red again at that vegetative stage, but then also look for carryover effects at the flowering stage. So we applied far red just for that 3-week vegetative period, and then remove the far red bars for the flowering stage. And what we found is there were very little carryover effects at the time of flowering. We did see a height increase. It was only significant for TJs. Fresh weight, dry weight, flower dry weight, and stem weight did not show statistically significant increases. And then there was a potential increase in CBD for TJs, but it was not statistically significant. So we have our linear regressions here. So height was significantly taller for TJs with far red at the vegetative stage and then harvested at the flowering stage. So the carryover effects at the flowering stage of applying far red at the vegetative stage only led to an increase in height for TJs and did not lead to other carryover effects. So our overall conclusions were that with increasing far red from our base level under these white LEDs was about 2 to 3%. And then depending on the experiment, we increased far red to 10 to 20%. It led to primarily increased plant height, reduced stem diameter, thinner leaves, and lower flower dry weight in some cases. So these would be considered shade avoidance responses, and then not particularly beneficial, right? If we are seeing less flower dry weight, ultimately, that is not a positive finding. And if a plant is taller in height, that may mean that we need to spend more time trellising it, or there's a greater likelihood of crop lodging. We did see differences between the two cultivars that we evaluated in terms of the degree of their response. So like almost anything that we study in plant science, it does seem to be a cultivar specific thing. And then far red above 2 to 3%. So our overall conclusion is we're not seeing particular horticultural benefits or useful benefits of having more than 2 to 3% far red. We didn't go below 2 or 3%, so I can't say what is the critical threshold, but it doesn't appear to be that there are commercially relevant benefits of having more than 2 to 3% far red, at least under the conditions similar to our experiment, which was sole source lighting under relatively low daily light integrals. And we always recommend that commercial producers do their own small scale trials with their cultivars and the other environmental conditions that they have in their settings. With that, I want to thank the Foundation for Food and Agriculture Research and the Hemp Research Consortium, and then Hawthorne that funded our work on this project. And then at Cornell, the Joss Rose Lab did our cannabinoid analysis, and Larry Smart's lab provided the plant material. And of course, my master's student, Brett Timmons, did all of the day-to-day hands-on work. So he's the true hero in this case. Okay, with that, I think we have some time for questions.
Yes, great. Thank you, Ricardo and Neil, for that awesome presentation. We do have some questions. So I'm going to start by going back to Ricardo's presentation. There were a couple of questions specific to that one. So first of all, what was the amount of water adjusted with the amount of light in the experiment?
Yes, thank you, Haley. Very good question. Yeah, so Christian measured water uptake per treatment based on pot weight and also volumetric water content using a moisture sensor. And the water was provided to maintain the same moisture content at each pot. So obviously plants were transparent and more due to the light, the high light treatments, then they were getting the adequate amount of water. So the short answer is yes, everything was water based on the need.
Okay, great. And what was the light spectrum, red versus blue, that you used for the supplemental lighting?
We use the pictures that you see in the images from current, the risk element light with the light spectrum, the PPR, which is 10% blue light, 90% red light. Yes.
All right. Going to Neil's presentation. Did you see Janet's G form anthers within the female inflorescence when grown under low light?
Ooh, good question. So, we did not see at least very much of this anther development specifically under low light. Our daily light integrals were about 17 to 20 moles, but it didn't seem to be a problem in our experiments.
Great. And what kind of results would you expect for the far red studies if the input light spectrum was broad? So something like sunlight instead of LEDs.
Yeah, so the LED spectrum is relatively broad. It's not exactly matching sunlight, but what it is it's a blue LED that's phosphor coated to spread light out over a broader range. So we did not use red blue LEDs. We use these like broad spectrum white LEDs. And then let me see if I could share that spectrum again. I'll just share my screen. But it is a fair question. The light spectrum effects that you see, you always have to ask what was the background light spectrum. All right, here is, so we had white LEDs where we get this peak in blue, and then we get another peak in kind of green to red. And then on the graph on the right, it was our far red peak.
Thank you. The next question can be to either of you. So do you have edema in cannabis like you do in tomatoes? Do you think blue light or far red light would improve this in CEA?
Yeah, I have seen edema or intumescence, kind of these blisters on the leaves. I feel like I've seen it in greenhouse conditions sometimes. I don't recall seeing it in these particular growth chamber experiments. So I know, yeah, and then Ricardo, have you seen edema at all in cannabis? And I haven't seen any literature about preventing it in the same way that UV or far red can prevent it in tomatoes.
I have not seen edema, Neil.
Mm-hmm.
Christian is in the audience, so he may be able to tell us if he saw edema, especially during the rooting conditions. But no, I have not.
All right. Next question, can you confirm there was no CO2 supplementation at any of the treatments mentioned, especially for the 0.21 grams yield per mole of photons?
Yes, no, that was ambient CO2, so highly ventilated greenhouse, and the CO2 was maintained in an ambient, actually measured at ambient. We didn't supplement it in the greenhouse.
Mm-hmm.
And I
Great.
should have mentioned in my experiments too, they were just ambient carbon dioxide, no supplementation. Although the effects of CO2 are well known for increasing yield.
Also for Ricardo, how did you calculate 10% moisture content of flower material?
So the flowers, so we had a group of flowers that were bone dry as a benchmark to see what is the full dry weight. And then the flowers that were kept for the experiment measurements, they were dry, you know, under 25 degrees C and 50% lattice humidity until we got to the weight that was included 10% of moisture. So we have a reference sample for that.
Okay. I'm sorry, we're having a thunderstorm here. I don't know if you can hear that, but we have some loud thunder in the background. Hopefully the power stays on.
We'll have to take it over if not. Yeah.
Yeah, that's why my room is so dark all of a sudden. Was there any study on end of flowering UVB use?
Sorry, what was that last part, Haley? Oh, end of flowering ultraviolet B. We do have some work in progress where we are looking at the effect of UVB light. There's kind of conflicting literature up to this point where there was work done in the 80s with medical variety of cannabis that found increasing THC concentration with a certain exposure to UVB. And then more recently, Yubin Zhang's group at the University of Guelph has tried more modern cultivars trying to very precisely match the doses that were used in the 80s. And they did not see good benefits. They just saw that UVB stressed the plant or injured the plant, and they had smaller plant size and not beneficial effects on THC or cannabinoids. And so currently through this FFAR project, our second series of experiments that we're doing now are exposing hemp plants to UVB for the last a couple weeks flowering. So our thought is maybe we could build up the biomass without having the stressful effects of the plant, and then see if UVB can prompt some of that secondary compound production.
Great. Next question, did you see any changes in sex expression when adding far red light?
We did not. I guess I should ask Brett specifically, but I can't remember any cases where he had to remove plants like hermaphrodites or males from the experiment. So I don't think it was for us it was not an issue. Yeah.
All right, well, that is all of the questions that I have. So thank you so much, Dr. Mattson and Dr. Hernandez, for presenting with us today and for answering these questions. And thank all of you for joining. The recording, as always, will be posted on our website and our YouTube channel for GLASE.
Thank you, everyone.
right.
Thanks for hosting
Thanks
us,
so
Haley.
much.
Have a good day.